First of all, thank you so much for the organizer inviting me to Lund. I'm very happy to be back here because for me it's been a place of change and evolution. I went to university here. During that time I also met a girl that is today my wife. So now it's like 4 children, 3 dogs, just 1 wife, but there we go. But it's also the city where I began my career in the life science industry. I took on a job for Gambro, selling dialysis equipment. And since then, I basically stayed true to that industry. And it's a great industry to be in. Now I find myself working with cancer diagnostics. I was appointed CEO only 2 months ago, so please bear with me if I can't answer all your questions. If I can't, I'll make sure to find out those answers instead. What we do is we can detect very, very early signs of residual or recurring cancers, sometimes years before you can find them on imaging. Pretty much to date, 31 years ago, my mother passed away in a breast cancer that relapsed and spread through her body. I don't know, obviously, if our technology could have meant any sort of difference to her. I do know that if we build this company and we can make our technology available to many more patients, others will benefit. Of that I'm quite certain. I took this job because I truly believe we can make a real difference to everyone with cancer, to everyone developing drugs for cancer, and everyone treating those with cancer. In short, what we set out to do is to unleash that insight that is necessary to make cancer finally a curable disease. Today's challenges in cancer care is late detection of relapse. With our technology, as I said before, we can detect relapse up to years before we can do it with, say, imaging. Uncertainty of treatment efficiency. Again, we use live biomarkers so we can see if any sort of treatment is effective or not within the hour. It's that short a time. Failure to detect remaining disease, say after a surgery, is there still some parts left of the tumor? We can do it with a simple blood test. What we do, or how we do it, is that we look at ctDNA. That stands for circulating tumor DNA. All cells die, as you know, and when they do, they will emit genes or parts of their DNA into the bloodstream. When cancer cells die, it's called ctDNA. So we find these ctDNA, and they exist only for a matter of hours before they sort of dissolve in the blood. So that's why it's a live biomarker. If we find it, there's something emitting it, and that is a tumor. This pretty much shows where we come in. When a cancer is diagnosed, there is an immediate treatment. We can see whether that is efficient or not. Then there is an intervention. So is there anything left of that tumor after the intervention? We can see that with our technology. Again, we detect relapses very, very soon. And when those relapses are being treated, we can monitor the effectiveness of that treatment. How we do it? Well, there are different ways of analyzing ctDNA. Sometimes you just look for one specific gene because that will be telling you whether you should be using one drug or the other with all this, personalized medicine. We do not do that. Or you can have panels that look for specific mutations that are normally found in, say, breast cancer. However, your mutation may have parts of what is known to be common mutations, or may have none of those common mutations. So there's always a high risk that they won't be able to detect ctDNA for your specific cancer. What we do is that we get the biopsy. There's always a biopsy taken when you're diagnosed with cancer. That biopsy gives us insight into your specific mutations. And those are the ones we look for. We don't just look for needles in a haystack. We look for very specific needles, the one that are your needles. As far as customers goes, we have two major customer segments, biopharmas and clinics. Biopharmas, we primarily focus now on the various clinical trial phases. So that's where we get in and we basically check whether a candidate is successful or not, or to what extent. That, of course, can save a whole lot of money because you can kill certain candidates quite soon if we find that, no, it's not working. We can also pre-select candidates to be included in those trials. So if you want to try your candidates on 200 patients, we can cut them down to 50 patients and still be as accurate and relevant. We're also currently validating our technology for preclinical trials. We require only a very small piece of blood, so we can do multiple tests on, say, mice, for example. And then finally, we also look at clinics because of them following up their patients, obviously, and finding relapses as soon as possible and monitor the treatment efficacy afterwards. But main focus today lies within the clinical trials. How we go to market is directly to the biopharmas or through the CROs. CROs, it's like going to a few to go basically to reach many. They already have existing customer connections with biopharmas, so obviously it makes sense for us to join forces with with CROs to be part of their portfolio. But we also approach already today biopharmas directly. Clinics, we can also approach directly or through certain labs. Again, the same logic applies, going to few to reach many. And finally, we can do kits, but that is not our core business. Kits are these pre-designed panels that you use to find cancer based on the most common mutations. We can do it. It's sort of like sometimes you need to have it in your portfolio, but we're not focusing on it, and we're certainly not going to be a manufacturer of those. We're going to design it and leave it for others to manufacture. There are such discussions ongoing. We had our first customer last year, so it wasn't due to anything to do with me, obviously. I've only been here for 2 months. But we came in at slightly below 1 million SEK. Today's forecasts look like we're going to come in around 4 million SEK, and it's perfectly viable to go for 250 million SEK by 2030. That's actually in the lower level of my expectations. The market is certainly there. What we're struggling with a bit is, within academia, it's a fairly well-known concept, ctDNA, its advantages. Whereas with some of our customers, they are knowing about ctDNA analysis, but not always about the pros and cons of it. So there's some education we still need to do. We do it fairly well, I have to say. Short-term plan ahead. We were selected by Business Sweden to be part of their Catalyst program with a focus on entering the UK. So that's our first market outside of the Nordics. So there's going to be a lot of things going on in the UK in the near future. We expect to sign the first customer later this year. Also, as we have ongoing discussions with CROs, an international one in particular, we expect that to be finalized sometime end Q3. We're going for a laboratory-developed test, which is needed to work with clinics. Today, when we work with clinics, it has to be research use only. With the LDT in place, we can work directly with clinics and have them use it as a decision tool. And we're preparing for additional market entries, as well as obviously working to get more biopharma customers. As far as geographic entries and geographical expansion goes, our philosophy is that we should really get a firm foothold in certain markets and not be thinly spread around the globe. So we're in the Nordics. We're going to expand to the UK. UK is good because they have very well-defined clusters of biopharma companies. Oxford, Cambridge, Manchester, London. Same thing with Switzerland, also kind of well-defined clusters. So that's where we're going next, and it sort of makes sense to go from there to Germany. After that, I don't know yet. We may enter the US. It's not been decided. We want to have a firm foothold in these markets. One differentiator for us is to deliver an outstanding excellence in customer experience. Therefore, we must have a firm foothold of a market before we expand elsewhere. We must always ensure that we can deliver that customer experience before we go anywhere else. It is possible to outgrow your abilities, and we don't want to do that. So to recap, we can discover cancer relapses sooner. We can provide relevant insight on treatment effectiveness, but also on dosage setting. I didn't mention that before, which is really important, especially when you treat children, to get the right dosage so you don't over-treat them, which will cause more harm. And it shortens the time for biopharma companies in clinical trials, which of course is a lot of cost saving and potentially also a faster route to the market. We have an excellent board of directors, a lot of good, relevant experience. We have a perhaps slightly more excellent team working inside Simsen. And with that, thank you so much.
Yeah. Thank you, Pontus. Is your technique effective regardless of cancer type?
Solid tumor cancer, so not leukemia, but otherwise, yes.
That was quick, so could you expand on how you came to that 250 million SEK target?
It's very hard to estimate the market potential because everyone trying to do so comes up with different numbers, and you never know whether it's just the analysis or if it's also including the equipment, etc. But I'm seeing the growth, I'm seeing the number of clinical trials in Europe. So yes, I mean, it's sort of, yes, it is guesswork, but it's also some intelligence included in that guesswork to look at the potential because of the number of ongoing trials, the upcoming trials, the international expansion, going into preclinical trials, and we already have discussions with clinics that are interested in working with us, even if it's just research use only. So, yeah.
All right, fair enough. Well, unfortunately, that's all the time we have. But thank you so much for your presentation, Pontus, and for answering the questions.
Thank you.
Thanks for being
here. Thank you for listening.